Hello, everyone, and welcome to this episode of Pod Save Chocolate. My name is Clay Gordon. I'm your host, and today we're going to be talking about the perfect chocolate, and more particularly, how to make perfect chocolate. Before I begin, I'd like to remind everybody that no matter where they are connecting from in the world, they can go into the live chat at YouTube or the comments in LinkedIn or Facebook and let me know where in the world you are connecting from. You can also let me know if you have any questions or any comments about any of the things that I'm going to be talking about. I'll get the shout outs, I'll get the comments and the questions up on the screen, and we'll be able to go through them today. And I'll give you a thanks for the shout out. And also a reminder that this episode of Pod Save Chocolate, like every episode of Pod Save Chocolate, is sponsored by the members of The Chocolate Life. And here is the home page of The Chocolate Life right now. You can go there. And if you want to join and support the work that I'm doing here, you can click on the green button in the top nav. It will say join if you're not logged in, as I am. There's always a free membership tier, but if you do have the resources to support the work that I'm doing financially, some of the levels of support also come with coaching and consulting hours. And you just go and learn more about that by going visiting the join button. And now let's get to the topic of today's episode. And which is making the perfect chocolate. So let's go and think about what that means. So one of the questions I have is like, how did I get here? So this is the post for today's episode. You can get there from the homepage. It's a run of show. I don't script these things. This provides me with the order of things that I've thought about talking about them in the run up to going live. But let's talk about how I got here in the first place. Why am I talking about making the perfect chocolate? And the answer to that question is, really, really quite simple. I subscribe to a number of newsletters, and this is one of them. It's called 1440. So I probably get, I probably subscribe to about 20 industry newsletters and a couple of general newsletters. So this is one, 1440, or there's another one called Charter. I also have a subscription to Google News Search or Google Alerts. And this is one that came in earlier this week. The title of it is Texas Redistricting, Soho House, and Perfect Chocolate. And you never know what's gonna be here. But I'm gonna scroll down here, and there is in the etc. portion. So let's actually join. So what is 1440? We'll give you a little bit more information. So 1440 is a subscription newsletter. It is a human curated look at news. And not only do they look at the news, but they provide some analysis. And I use it just to keep on top of topics in business and finance and science and technology, health and medicine. As you can see, there are topics here. I use it in conjunction with a service called Ground News to keep track of things. And it provides me, which you generally keep on top of what it is that's going on, but sometimes I find ideas for topics for episodes. And separately, and this came into my feed this morning, so it's not something that was part of my thinking for this, is, excuse me. So separately, what came in is I received another one of the newsletters I'm subscribed to is called Cocoa Radar. And Cocoa Radar is written by the ex-managing editor of Confectionery News magazine, Tony Myers, somebody I've known in person since at least 2017. But the current issue of Cocoa Radar, it arrived this morning, just a couple of hours ago, is on this topic, Unveiling Colombia's Secret Sauce, How Microbes Are Rewriting Fine Chocolate Flavor. And is that true? So he doesn't go through a whole lot about what's going on, but they have this exclusive white paper, which the people at the Cocoa Radar team have written on top of what I believe is this article that showed up. And the article that showed up is this one from Popular Science. So how to make perfect chocolate according to science. Now, if we go back and look at the post for today's episode, I've just taken a screenshot of the top of this, how to make perfect chocolate according to science. So what it says here, the excerpt, the subhead is, cacao starter cultures may lead to a chocolate fermentation renaissance. So number one is we're now, okay, so this is popular science. And we have to consider who the readers of popular science are and what it is that they're looking for. So I think the magazine, especially online, has become more clickbaity, less in depth than the print publication, which I used to read on a regular basis when I was 12, right? I haven't been a regular reader of it as an adult, and certainly not in the internet age. But one of the things I noticed right off is that there is this conflation between how to make perfect chocolate and chocolate fermentation. What they're really talking about is they're talking about cocoa fermentation. So cacao starter cultures lead to chocolate fermentation. That gives you an idea just how much the author understands about what it is that they're talking about. But the audience here is popular science, so they're taking complex topics. It's probably not as sophisticated an audience as perhaps Scientific American is, but right, it is aimed at somebody could feel good about having a top level knowledge, if not understanding, about an interesting topic that they can start talking about to people. And so my question is, how well does the article actually reflect the topic, which is, are we going to learn how to make perfect chocolate according to science by reading the article? And the spoiler alert is no, right? The article isn't going to talk about how to make the perfect chocolate according to science, right? And I go through it. Now, there are a couple of reasons why that is the case. One of which is the article is 700 words long. There's not a whole lot you can learn in 700 words. There's not a whole lot you can convey in there. But we can take some top line points that somebody might be able to, somebody who's not really understanding the topic or doesn't have a huge amount of information about the topic, let me make this a little bigger so it's a little more legible for you. So there are a couple of things we might learn. That natural fermentation is unpredictable. That's true. Controlled fermentations are controllable. That's in the definition. So it's a tautology. If you control the fermentation, it's controllable. The more we know about the variables involved, the more control can be exerted. That makes an enormous amount of sense. But ultimate control includes using a tailored, and by tailored I mean not naturally occurring, mix of yeast and bacteria, and what the research article that this is based on called microbiota. So this is the article. So again, here is the article, and we want to go figure out what it is that they're saying, right? So the major part of this is this link to this article in Nature Microbiology. And I'm actually going to spend the most of my time in this episode going through the article in Nature Microbiology. And hopefully what I'll do is I'll give you a little bit better understanding about how to read a research paper. And I think that's the point here, because we're not actually going to learn a whole lot about perfect chocolate here. We might be able to extract some understanding of process and methods from the article about cacao fermentation and think about how we might apply them to chocolate. But the important thing about understanding the clickbait headline, which is what this ends up being on this article. So the clickbait headline on this article is how to make perfect chocolate according to science. And it turns out we're not going to learn how to make perfect chocolate, because at no point in this article does it talk about anything related to the chocolate making process. Now, this article in Popular Science doesn't talk about roasting. It doesn't talk about bean selection. It doesn't talk about cracking and winnowing. It doesn't talking about roasting, which I may have mentioned already. It doesn't talk about refining. It doesn't talk about conching. It doesn't talk about anything about the process of making chocolate. All it does is talk about cocoa bean fermentation. But there's a lot we can learn about cocoa bean fermentation. I just don't want anybody to think that they're going to go to this article in Popular Science, and by the time they end up reading 700 words, they'll know how to make perfect chocolate. Not going to happen. It doesn't happen. It's hugely clickbait. So I want to go and focus on that. But there's a little bit of history. And it's interesting to know, whenever you're doing something like this, is to find out okay, where are they linking to? So they've got this link to another article on popular science, right? So you can learn a little bit. This is about 3 years old. So, that's an interesting article here to learn. We've got another article about Mesoamerican cultures and their histories in cacao and culture. And it turns out that this website has got a whole lot of really, really interesting information. So what I will do is I'll take a link to this, but this free curriculum on and links about cacao and chocolate has got a bunch of interesting information just in terms of general knowledge about cacao and chocolate culture and history. Right. So, and if you're interested in putting together a curriculum on teaching cocoa and culture, cocoa and chocolate in a classroom, it's a good place to go started. There's also an article about cultures using cacao as currency, and I so get to that particular video and show you the ad, but it shows a YouTube video. So go and take a look at this and figure out what's going on, right? And understand that what it is that the article really talks about. And before we get to the article on the microbiology, but the author is breaking down the differences between spontaneous and controlled confirmed fermentation, right? They're saying that farmers have little influence traditionally, have little influence over which microbes dominate or how the fermentation process unfolds, which is true and untrue at the same time. It is true that farmers have little influence. And I would say that farmers have no influence over which microbes dominate, right? But they do have influence about how the fermentation process unfolds, right? They can decide if it's going to take 2 days or 4 days. They're going to decide if it's going to be heap fermentation, which has its own characteristics, or box fermentation or tray fermentation. There is a fair amount of control that an individual smallholder farmer can exert over fermentation, but they're not generally going to spend any time thinking about what the microbial, the microbiome of the cacao fermentation pile is. So it says, as a result, fermentation and thus the flavor and quality of beans varies widely between harvests, farms, regions, and countries. And this is absolutely true. But we get to that point and there's nothing, they talk about beer and bread and cheese, but they don't talk anything about, they don't talk anything about chocolate production. So we're not going to learn anything about that. But let's go and take a look at the article. Right? Take a look not at the article, but at the research paper. And what I'm going to do is I'm going to open up the research paper in a different browser. So the original browser I was showing is called Arc, from a company in New York called The Browser Company, which is my day-to-day browser for a bunch of reasons. But this browser that I'm showing you is called Dia, which is made by the same company. So it's a different approach to a browsing experience, but it's tailored towards somebody who is doing research on the web. And I'll show you about that in a little while. But let's talk about a little bit about what the article says and how we might go through it, and then what we might be able to learn from it. And again, if at any point you can go into the comments and say, hey, Clay, tell me a little bit more about that, right? Or you got a question, you got a comment, just let me know and I will get it up on screen and I will answer it. So this is the title of the article that the Popular Science, this is the title of the research paper that the Popular Science article refers to, which is a defined microbial community reproduces attributes of fine flavor chocolate fermentation. So we're actually fermenting cocoa to reproduce flavor characteristics that lead to fine flavor chocolate, basically. Always, if you can, go and look through sort of the first authors or the author list. So I don't know anything about David, who's the primary author, the first author on this, but I'm looking through the list, and Darren Suka is a name I know from the University of the West Indies. He's very heavily involved in the Cacao of Excellence program and the Heirloom Cacao Preservation Project. And also a name I know from the author list is Padmanathan Umarharan, who's also at the University of West Indies and is a colleague of Darren Suka. So there's some interesting names that I know here. I can go and click on one of these links, and it'll actually give us the where the authors are, and if we want to look at other articles that these authors have written. Start off just taking a look at what is the structure of the research paper. There's an abstract. We've got the main area, the results, discussion, methods, data availability, code availability, author, all this kind of stuff. So just take a quick look, and we can jump through the article or jump through the research paper using this, a list of figures, and then we have the footnotes and references that we can go and look through. So all of that is there, but the abstract, what is this research paper telling us about? And what it says is that cacao bean fermentation is a spontaneous process. So what does that mean? Because they don't describe what spontaneous means in the context of this. So the cacao pod exists in a natural environment. And in the natural environment, there are microbial colonies, yeast, bacteria, fungus, that exist naturally. And that yeast and the bacteria and that fungus are on the leaves and on the outsides of the pods. And so when somebody goes and harvests the pods, they're going to get those yeast and bacteria on their hands. And when they open up the pods, they're going to put their hands into the pulp when they strip the beans off the placenta. And so they're going to inoculate the pulp with the microbiome, with the yeast and bacteria and fungus that are in the naturally occurring environment. And therefore, fermentation is going to just kickstart. The moment you open the pod, right, that the yeast and bacteria, which are also in the air, are going to settle into the pulp, and fermentation is going to start almost immediately. So that's what spontaneous means. Right? It's going to involve interactions between abiotic, and abiotic are non-living aspects of this. So they're going to be the temperature, the sunlight, other things that are in the environment that are not living, right? And biotic, which are going to be yeast, bacteria, fungus, other things that are living that are in the environment, and that those things contribute to the final flavors of chocolate. And you know what? Let me go and get this a little larger. And right, that makes sense. So understanding the underlying interactions could enable desired flavor profiles to be reproduced under controlled conditions. So what this means is, if we know what the abiotic factors are and the biotic factors are, and we can control them, what we can do is we can control the output. We can control the results of fermentation. Right? And in general, the more we know about the abiotic and the biotic factors, right, and the more we have controls and constraints around them, the more reproducible the results are going to be. That makes sense. So what they're doing is they're saying, using bean fermentation from farms in Colombia, the researchers established that pH, right, so the acidity, the temperature, and the microbiota composition, the microbiome, the environment, including both bacteria and fungi, influence key factors that contribute to the flavor of premium chocolate. Furthermore, what they say is that using a defined, so a tailored and controlled and metabolically competent microbial consortium. So what they mean is that they can go in and select particular strains of yeast and particular strains of bacteria and particular strains of fungi, right? It becomes more feasible to replicate fine flavor attributes of chocolate under controlled conditions. And according to the research, this hypothesis was confirmed, right, using omics, right? So we'll get to if you don't know what omics are, we'll talk about what omics are, metabolic networks, and a trained tasting panel. So the results of the research provide the basis for the design of fermentation starters, so tailored starter cultures that you would inoculate a fermentation pile with in order to be able to drive it in particular direction to robustly reproduce fine chocolate characteristics. So that is the abstract, that is the hypothesis, that is the direction that the research is going to go. One of the great things about an article like this is going and seeing a bunch of related research. So this research, these 3 articles are not necessarily part of the references that are in here. They may be, but they may not be. But if you want to, it's really interesting to go and take a look at this. And even if you're not going to just read the abstracts in order to figure out what it is that they're saying. But you have links to lots of other research. But let's go, all right, to the next stage. So the main part of the article. So it talks about the production of fermented foods, such as chocolate. Chocolate is not directly fermented. Cocoa beans are fermented as an ingredient. We directly eat the milk that is fermented. So there's chocolate is fundamentally different from wine and beer and spirits and cheese, at least in my estimation, right? But the production of fermented foods relies on the metabolic activities of microbial communities. It relies on fermentation. So members of the community, so the microbes that contribute to fermentation transform the raw scrub state, cocoa, right, into the precursor for chocolate production. That makes an enormous amount of sense. We go through here. It says cocoa bean fermentation takes place on cocoa farms. Typically, it does take place on farms. Within wooden boxes, baskets, or heaps covered with various materials to regulate environmental temperature. That is very true. The farmers are often members of cooperatives, and so sometimes the farmers deliver the wet mass to an intermediary or to a cooperative to do the fermentation. So this is in contrast to other fermented foods, such as wine, cheese, beer, where microbes are often intentionally introduced or controlled. The microbial communities in cocoa fermentation assemble spontaneously. They're present naturally in the environment. I don't know that they assemble in an Avengers Assemble kind of way. They're just naturally occurring in the environment with minimal human intervention. The only human intervention might be if we're using pesticides and herbicides and fungicides that are going to destroy the balance, right? But otherwise, if I'm not applying agrochemicals, there's no human intervention in what the microbiota community is going to look like. So what it says is variation in the microbial communities across different locations are expected. That's true. And this can influence, not only can influence, it does influence the quality, chocolate, and flavor, the chocolate quality and flavor from different cocoa origins, which is something that we all know. I want to stop right here and point out that this research was done over the course of the last several years. I don't know when it was submitted for publication, but it was published, recently. The date on this is earlier this week. Right? This is not new. What is it they're talking about is not new. We have known that controlling fermentation and inoculating fermentation piles with tailored cultures is a thing. And we've known it for quite some time. And we can go to the references and we can see, here's one from 2004, right? Schwann and Wheels, which says the microbiology of cocoa fermentation and its role in chocolate quality, right? The functional role of yeast, lactic acid bacteria, and acetic acid bacteria in cocoa fermentation, flavor chemistry. If we were to search for the word inoculation, we'd figure this out, right? So it's the notion that we can control fermentation by controlling aspects of it is nothing new. And in fact, this is something that I saw firsthand working with Zoe Papalexandratou. And Zoe is mentioned in 2 of the papers in the references here. So you can go search for PAPAL and you'll find the papers that research here. You may be able to research them. But I met Zoe through Ingeman in Nicaragua in 2014, and I organized a series of fermentation boot camps. So people who are interested in what Ingemann was doing and interested in their technified approach to fermentation could go to Nicaragua, spend a week, and 3 to 5 days were spent on cocoa farms and at the processing center where Ingemann did all their fermentation, where we went through a lot of the steps that are talked about here. Now, that 5-day boot camp was not for microbiologists, it's not for people who had a knowledge of biochemistry or microbiology or genetics and what sequencing or genomes and omics and all that kind of stuff. So it was aimed at chocolate professionals, not necessarily scientists and chemists, right? But one of the things that I learned working with Ingemann back down in 2014 is that if you understand what the microbiology of the natural environment is, so if you know what the dominant yeast strain is, right, and you know what the composition of the yeasts are, because there may be 3 to 5 yeasts which are common everywhere, but one will dominate in one area and another one will dominate in another area, and the ratio of the yeasts will differ from one part of the country or from country to country or from one part of the world to the other. But if you know that and you understand the temperature and you understand temperature, so you're keeping track of temperature during fermentation, and that you understand changes in pH not only of the pulp, right, during fermentation, but also changes in the pH of the cotyledon, the bean itself. So if you understand those things, what you can do is you can control the outcome and you can be consistent. So for example, when you open up a bunch of pods and you put them into a fermentation box, you want to know, how much, what the sugar content of the pulp is, what the pH of the pulp is. You have an understanding of what the genetics are already. At least that's what Ingemann did, right? And understanding the starting conditions, what you can do is you can keep track of the temperature and you can understand pH. And then by keeping track of the temperature and the pH, what you can do is you can determine, the primary phase, what people might think of as the anaerobic phase of fermentation is done. And now we need to start the second phase. We need to introduce oxygen to the pile. And then what we're going to do is we're going to keep track of temperature and pH of the pulp and the cotyledon until it gets to a certain point. And by understanding what these things are and controlling them, you can do a really good job of making consistent quality cocoa, in the sense that the distribution of fermentation is going to be the same from harvest to harvest to harvest, from batch to batch to batch, from year to year, right? So that the buyer will know that, hey, the cocoa they're going to get is going to be consistent fermentation. So if they have a roasting profile, they know that they can use the roasting profile to make a chocolate that they already know how to make, which is what people want. Consistent quality. Whatever the quality is, it needs to be consistent. And this is something we've talked about a lot here on The Chocolate Life over the course of the last couple of years. And this is what this first part of the article is talking about under main. It basically says the more we know about this, right, the better a job we can do is controlling it, which makes logical sense, right? But they do talk about a high level. So unfortunately, I can't share the highlights with you. If I share the link to this article, this is just in my browser. So, but there are a couple of things. So changes in temperature and pH inform the progress of fermentation, which is something that I said that I learned, working with Ingemann and specifically Zoe Papalexandratu, whom I then hired to work on 2 projects with me afterwards, with a big one in Mexico. So changes in temperature and pH inform the fermentation progress. All right. So if we keep track of temperature and we keep track of the changes in pH, we can determine when essentially fermentation has stopped and we need to start it again, right? So going from the primary to secondary fermentation, we can keep track of these things are primary indicators. Okay. Again, something that I knew, a decade ago. It's been 11 years now, but it may be new for people. And this, I think, is where this research paper has done something really, really useful, is that it goes into amazing detail about the process. So they've gone and taken a look at lots of other research, and they've said, let's create a hypothesis and a research process which will enable us not just to confirm what it is that we already know, but to give us an idea about how to control things, right? Which is interesting, right? So, right, so we go through and all sorts of also, if you're a graphic person, you can go and take a look. But beneath each one of these sets of figures is this deep explanation, right? And so this is mainly of interest to scientists and statisticians who are going to look at the methodology and understand what steps were taken to achieve a particular result, and then how are the data analyzed in order to reach a level of statistically significant confidence. So how can we know that we can rely on the outcome of the results that we're doing? And I'm not going to go through it in great detail. If you don't know what these things are, I'll tell you why having a good browser is important. But they talk about how they did it and all this other kind of stuff. So that's interesting, right? So changes in changes in pHa and temperature, monitor the progress. They can tell where to make major changes. Changes in the microbiota composition drive bean fermentation. So what that says is different combinations of yeast and bacteria and fungi will change fermentation. Seems like it's straightforward. But again, they go into great detail about what that is. Fermentation characteristics shape chocolate flavor profile. So what this says, if we go through a particular fermentation, the flavor precursors that are in the cocoa beans when we get done will be different. And so we can take the same beans, ferment them different ways, and we'll get different chocolate flavors, which is something that Ingemann does. They have this one genetic variety, which is called Chuno, and I think they have 5 or 6 different fermentations which deliver consistently different results using this one genetic variety down in Nicaragua. But here they're talking about cocoa beans in Colombia, in Huila and Antioquia, and other parts of the country. And if you want to get into the real deep dive into what this is, you can go and read this. But they talk about the DNA analysis, the ancestry analysis of cacao plant samples, and great detail, more than I want to get through. But they analyze temperature and pH kinetics to test, and they go and just, if you've never read a research paper before, right, I recommend that you go read a paper like this. You are not going to understand a lot of it unless you have a statistics and or a biochemistry background. Chances are you're not going to understand a lot of what it is that they're talking about. But go through and read it and figure out what you can. So highlight it along the way, like I have. Get a web highlighter and do this, and try to understand what they're saying. And I have some experience reading these things, and so, but go take a look at it. Abiotic, which are the non-living, and biotic, living features are linked to sensory attributes of cocoa liquors, which makes an enormous amount of sense. So the amount of sunlight you get, the temperatures, weather, for example, what the chemical composition of water might be, things like that, rain, things like that, they're going to have an impact on the sensory attributes of cocoa liquor once the cocoa beans have been transformed into liquor. That makes an enormous amount of sense. And here you can go into as much detail as you want to understand what's going on. And here's a description of how they went and arrived at the graphics that they developed. So flavor is linked to fermentation, abiotic and biotic signatures. Okay, they're restating, right, what they've already talked about, but they're going to go through and they're going to say, how did we make this correlation? Makes an enormous amount of sense, right? Defined microbes reproduce fermentation and metabolic profiles. So this is where things, I think, get more interesting in this research paper, at least for me, which is what they're doing is they're taking a look at certain populations of yeasts, bacteria, and fungi, right? And they're going to try to determine them. And they're going to say, okay, what do we need? I mean, what do we need in order to be able to control the to direct the fermentation to get the kinds of flavor profiles that we're looking for. So they're using metagenome assembled genomes, right? Using shotgun sequencing data of bacteria and fungi associated with fermentation. You can go and look at what that means, right? Then they talk about that. So the metabolic characteristics of the microbial communities present in natural cocoa bean fermentation can be condensed into a reduced number of microbes. So what they're saying is that some microbes contribute significantly to a fermentation and some don't. And so if you want to have a tailored culture, what you want to do is you want to use the ones that are significant contributors to generating the flavor profile that you're looking for. And you can go and take a look at this. So, I've highlighted many aspects of this in green here, but you can see that just by reading the headlines, you can get an idea about where the research is going, even if you're not going to go into the great detail of going and reading the words. So a microbial consortium replicates desirable traits in cocoa fermentation. So what they're saying is we can pick a subgroup of all of the microbes, right? The microbiome. We can do a condensed version of it. We can select from it, and we can drive the flavor development in a specific way. Right? And less complexity is going to be easier when it comes to create starter cultures, which makes sense to do this. And so using synthetic community dropout experiments, what they're saying is, what does that mean? So the synthetic community dropout experiments, what they're doing is they're saying, let's go and do a fermentation using this selection of the yeast, bacteria, and fungi, the microbiota. And then we're going to do another version, but we're going to drop out one of them. And then we're going to do another fermentation, and we're going to drop out a different one. And so they're going through a stepwise process to understand how each microbe contributes to what aspect of flavor. So that's what this talks about, right? And then they talk about how they selected farms and how they did fermentation on the farms and how they monitored temperature, pH, and color bean analysis on the farm during fermentation, how they did sample collection. So this is more about process, which may or may not be interesting to you. But what it says is all if you want to get an idea about what's going on, just reading the headlines is really, really helpful. All right. So, and if you really want to get in there, you can talk about the specific pieces of equipment they used. You don't want to know, for the most part, what that is. So why would they use an Oxford nanopore technologies, whatever, right? Not really, really important to this, but if you want to know, you can figure this out. But here's where I want to go in and talk about how having a good browser, and I'm not compensated for this. This is not by any means sponsored, but it's one of the reasons why I have been on the beta test program, both for the Arc browser and the Dia browser, is it's hidden by my little CocoaPod up here in the upper right-hand corner. It's hidden by the CocoaPod, but there's a little button up here which says chat. And by clicking on chat, I'm now talking to an LLM, a large language model. I believe it's ChatGPT. I don't remember which model it is. There might be an option to change which LLM I'm working for. But also in Dia, what I have is the ability to go through and I can say, remember things. So personalized answers with memories, just keep track of things. And this is keep track of conversations in all open browser windows and all chats, not just the one I'm in, which is interesting. So I can also personalize the answers using instructions. So I can talk about, who inspires me, how do I digest information, how do I want Dia to write. So I have all kinds of control over how Dia responds to me, which is cool. So I can say I'm a professional researcher, I'm a journalist, right? I want something which is conversational, or I want something which is technical. And so I can get Dia to do that for me, which is not unusual. If I go to Claude or I go to ChatGPT or I go use Gemini or the other LLMs, I can do this. But what's really, really nice is I have this chat pane right next to a document. And in this particular case, I'm going to scroll to the top here. In this particular case, I have asked I've asked DIA for a summary. So I have a shortcut /sum, which is for summary. I've asked it to summarize this document. So it knows that this document is open. And I've asked it to summarize this document. And it just knows that. When I click on, when I click on here and click on chat, it knows that I'm talking about this document. So it makes it easier. I haven't left the context. Don't have a browser window open. Then I have a different window open to talk to ChatGPT or Gemini or Claude. I mean, I'm in the same context and I can see them back and forth. This is really nice, not having to change context, right? So it knows that when I ask to summarize for an audience of adult college-educated chocolate lovers who are not experts in microbiology or chemistry, when I do this, I say summarize for this audience, this article, what it does is it writes a summary, right, of the article. And it says the research investigates how microbial communities involved in cocoa bean fermentation shape the flavor of chocolate. Oh, by the way, I've told it that I want to use American English and American grammar rather than English UK spelling, right, and UK grammar. Even though the article is written OU because the lead author is at the University of Nottingham in the UK, I want it in US English spelling and grammar. So traditionally, cocoa fermentation is spontaneous with microbes from the environment. So that ambiguity around spontaneous and assembly has been removed, right? It may mean things in specific context to researchers, but it's not necessarily understandable to somebody who's not a trained in this area. So the study explores whether a carefully selected defined group of microbes can reliably reproduce the complex flavors associated with premium fine flavored chocolate. Cool, right? So I've got in this one paragraph right here, right, all of this kind of stuff summarized, and it's done a good job. It's done a really good job. So the key findings, it goes through key findings about fermentation and flavor development, microbial communities, abiotic and biotic markers, microbial diversity, geographic influence, defined microbial consortium, the validation process, and the implications for industry. It did a really good job. Why it matters. Okay. This is not necessarily a summary for the article. This is not in the article itself, the research paper. So why it matters for chocolate lovers: consistency and quality, flavor diversity, traceability, and innovation. I don't think that chocolate lovers really care about innovation at this level, and traceability. This approach could help chocolate makers better trace and reproduce the flavors associated with specific regions or styles, supporting both tradition and innovation. So now that I think about it, one of the things that my friend Zoe Papalexandrandu has been doing with MOCA, so the maximizing opportunities for coffee and cocoa or in the Americas, one of the things they've done is they've developed a flavor map. And what they said is that certain beans with certain kind of fermentation in certain areas of Latin America, when you take those processes all together, all those variables all together, they can create flavors in chocolate that are very, very close, right? But the beans are grown in a different country using very different fermentation and post-harvest processing techniques, right? And so this is interesting. If by understanding and controlling fermentation and drying, what we can do is we can have two cocoa beans grown in very different places, right, that can be used to produce chocolate that tastes very, very similar or maybe identical. Why is this important? Well, it may be that I'm a chocolate maker and I want to produce chocolate from 20 tons of beans, right? But I can only get 5 tons of beans from one place. But there are other places where I can get beans that are essentially identical in terms of the output that they will generate. And I can go make up the rest of my 20 tons by going to these other places and by buying beans which are can produce chocolate, which has flavors that are very similar, which I think is really, really cool. And it's one thing that the MOCA project has done, that Zoe has done with the MOCA flavor map. So, and then it does a summary of the reference content. So then I'm asking it, I looked at the references, right? So the that are down here. So what are the other papers that are referred to? So I'm going to click on one. So I've looked at these other papers, and I'm going to go, and if I wanted to, I could go search on the word patent, and oh, there are patents, right? So non-Saccharomyces commercial starter cultures, scientific trends, recent patents, and innovation in wine, right? So they're obviously looking at patents in wine. Are they applicable to chocolate? So then what I asked DIA to do, again, is research. It has this document in its brain, so in memory. So I asked it to research, and so research here is another shortcut, and it means something in the DIA context. So there is a skill which is associated with this. So when I use the shortcut research, there's a set of prompts that it follows, right? So, and this is the shortcut, which is really, really nice. So research the history of research and patents using inoculations of tailored cultures in cocoa fermentation. So I say, what do we know about this, right? So it gives me a couple of key early studies. These top three are all referenced in this in the references section. Swan and Wheels, DeVoust and Leroy, and Camus are all in here. But going back to the 2010s, so 15 years, we talk about starter culture development with references to particular studies, patent trends, recent developments, current state and outlook, and then they give us some specific references that we can go and look at. So then, right, one of the things that DIA enables me to do is I can go highlight some text, like highlight. So I was able to highlight the text and do research on the highlighted text. And so it gave me some patent history and trends, tailored cultures and cocoa fermentations. And also gave me some additional reading to do this. But then I wanted to ask it a question, which I think is fundamental to the entire process, the entire line of questioning. And it's one of the things which was not touched at all in this Cocoa Radar article. So I'm not a premium subscriber to Cocoa Radar, and so I can't download their white paper when it comes to fermentation. But what I've done is I've read the research article and I've used an LLM to ask it very, very specific questions. And so I don't know what the insights team at Cocoa Radar has come up with that's going to be different from this, right? But what they're talking about here is interesting. Colombia's secret sauce. Well, first of all, it's not Colombia's secret sauce, right? This is a bunch of researchers, many of them in the UK, but from around the world, right? Right, talking about how microbes are rewriting fine chocolate flavor, and they're not, right? This headline. I'm sorry, Tony, if you're reading this or watching this, is that the microbes are not rewriting chocolate flavor. This is research. This is stuff we've known for 2 decades or longer. Right? So there's nothing about there, and there's nothing specific about Colombia. What I would do is I would go and talk to you about the work that Ingemann had been doing in Nicaragua and the experience that Zoe and a bunch of other people have had in production. So this is not theoretical. They're actually doing this production. They're producing tens of tons, if not hundreds of tons, of cocoa every year using these controlled fermentations, even if they're not using inoculated, tailored cultures, starter cultures in order to do it. So another clickbait headline. Sorry, Tony. Right? This is not a good reporting of the research paper. Right? But they talk about this from the sustainable farms of Colombia. The alchemy of cocoa and its transformation is a science that conceals many of its secrets. The alchemy of cocoa and its transformation into rich, indulgent chocolate is a science that conceals many of its secrets. I don't know what that means, right? Now, new research is uncovering some of those microscopic secrets behind your favorite chocolate bar, giving artisan chocolatiers and farmers alike a fresh new perspective. So by fresh new, we've known about this for more than a decade. I mean, and it may be 20 years or longer. A team from Nottingham has identified what they call the secret sauce of chocolate, a specific blend of microbes that can transform cocoa bean fermentation unlocking floral, fruity, and citrus tasting notes while reducing bitterness and astringency. So, number one, I would agree, right, that if I wanted to take a look at bulk cocoa, that reducing bitterness and astringency is a desirable thing to do. But what the insights team at Cocoa Radar has failed to appreciate is that if I am a big chocolate maker, so I am a business-to-business chocolate maker by Barry Callebaut, I'm one of their customers, right? If I am Ecamm or IRCA or Cargill or any one of these companies that are doing very, very large scale processing of chocolate, right? I have my recipes locked in, right? My customers expect my chocolate to taste a particular way. Somebody who's been buying a Hershey bar for the last 20 years, Hershey's milk chocolate bar, all of a sudden doesn't want fine flavor characteristics in their cocoa, right? They don't want fruity and floral in their cocoa. They want their Hershey bar to taste the way it always has, which is slightly sour, because that's what it is that they're used to. So, number one, the notion is that there is a huge demand for fine flavor cocoa in the world is completely overblown. At the moment, it's much less than 5% of the market. If there is a use for it, industrially, it's primarily as a blend. If we take a look at what's happened over the last year or two, particularly with the rise in the price in cocoa, is that Cacao Berry has dropped, a subsidiary of Barry Callebaut. Cacao Berry has jettisoned a lot of its single origin line. Even Guittard is no longer making the same line of single origin chocolate bars. Barry Callebaut itself has completely stopped its second generation because it's just too expensive. I mean, the idea that chocolate makers are going to pay more for cocoa, which has been processed this way, right, so they can have flavors that they don't need, makes no sense whatsoever. Another thing they don't talk about, and this is something that I've spoken about with other fermentation researchers, is do we want to focus on fermentation, which can, and while it's reducing bitterness and astringency, also raises the nutrition profile of the cocoa beans. So do we want to push more fine flavor? I don't think so, right? The market is not demanding, especially with where the price is right now, more for flowers, more fruit, more citrus, right? Right, in its cocoa beans. They want consistency. They want to know that if I had this flavor last harvest, I'm going to get the same flavor this harvest. I've been making this chocolate for the last 20 years. I want to be able to make this chocolate next year. I don't necessarily want more flavor in my cocoa beans. And this is something that is, I think, a lot of people don't understand. And it's not talking about in this article, right? So by analyzing fermentations in Colombia, researchers discovered that Antioquia's microbial community led to a more generic flavor, similar to bulk production beans. In contrast, beans from Santander and Huila delivered refined fine flavor notes reminiscent of prized Madagascan cocoa. This could be interesting, right? So if you are thinking about importing Madagascan cocoa beans, transporting them, and you can get beans of the same flavor from Colombia, that could be interesting because it may be less expensive for you to get cocoa beans from Colombia. That I can get the same flavor profiles, right, from Colombia beans in particular parts of the country. That's interesting. But there's an aspect of this that I do want to talk about, which goes back, let's go back to the other browser, which is one of the questions I asked was, what is the cost of implementing the use of tailored microbial cultures for cocoa fermentation in African cocoa producing countries? So it might be the same worldwide, but I'm thinking about what would it cost to implement this just in the major growing countries in West Africa, particularly Cote d'Ivoire, Ghana, Nigeria, and Cameroon, right? So it's going to, once you have developed, I have no idea how much the research cost. I have no idea how much this research costs, but the cost of producing or purchasing microbial starter cultures runs from a few dollars to several dollars per batch. So it doesn't say how big a batch is. So there is the cost of starter culture production and supply. but requires investment in basic microbiology infrastructure, which probably isn't cheap. Um, I don't necessarily, are there cold chain requirements? So if I've got this in a laboratory, do I need to keep it cold all the way to the farm so the farmer can use it? That's another issue. Um, there are training and extension services costs. We have to adapt the processes and then ongoing input. So regular purchases or propagations of the cultures, right? So I need to change the way things are done. I may need to invest in new fermentation boxes and other kinds of stuff. So starter cultures can cost $10 to $50 per ton of wet beans. Right? So this comes from estimates. So this is interesting. So if I've got $50 per ton of wet beans, that's 400 kilograms of dry. So the 50 becomes, so it becomes another 90, double it. It's almost $200 per kilogram for dry cocoa beans for starter cultures. Is the market going to pay another $200 per kilogram for starter cultures? I've got training and extension of 2,000 per cooperative community. If I've got 1,000 farmers in a community, it's going to cost more than $2,000 to train all of my farmers in that. So I think that's a low number. Equipment upgrades. I got to buy fermentation boxes if needed for improved control. This is all about improving control. So yes, I'm going to have to start fermenting in cascades rather than in heaps. Inoculating a heap makes very, very little sense. So there, then we've also got transportation costs from on the farm to centralized. So these numbers, cost per ton, decrease with scale due to bulk purchasing. Some commercial operations in Western have reported total incremental cost of $30 to $100 per ton for full implementation, including training and monitoring. I'm skeptical that that number is actually comprehensive in terms of what's going on, just given the cost of starter cultures, right? So maybe there's a way to bring the cost of starter cultures down. But at this point, right, in Ghana, where the farm gate price is controlled by the government, by COCOBOD, right, who is going to absorb that $100 per ton cost, right, for improving the quality, right, of Ghanaian cocoa? When people are buying cocoa from Ghana, they're not necessarily looking for fine flavor cocoa. They're looking for bulk commodity flavors. Again, Ghana is the reference flavor for cocoa in chocolate. That's not what they're looking for. And will the government raise the farm gate price significantly enough so that all of the extra work and all the extra cost is captured, right, in the increased price that gets paid to the farmers and cooperatives? I don't know the answer to that question, but it's really important in a country like Ghana, where the farm gate price is controlled, Togo, where the farm gate price is controlled. It's in Nigeria and Cameroon, it is more entrepreneurial, but there's still the question of who is going to pay for this, right? And ultimately, the consumer is going to pay for this, right? But if I'm adding $100 to the cost of cocoa at the farm gate, what does that mean for the world price of cocoa? Who's going to capture that? Where is the profit going to be made? None of the discussions about this technified fermentation, right, discuss what is that going to do to the price. And this was a huge discussion that I was having with Ingemann in 2014 through 2017 when I was working with them. How are they going to justify the cost that they were charging, the price they were charging for their cocoa? And this was before the market went crazy in terms of bulk cocoa, commodity cocoa prices. So this is an interesting thing. So it says is the added cost is often, but not always, offset by higher bean quality, better fermentation consistency, and access to premium markets, which can yield price premiums of $100 to $400 per metric ton for fine flavor or certified beans. And this is, again, where it takes experience and background in history, which is experience, to go, this is a case where the LLM doesn't have enough information to be able to make a good, give a good answer. So the barriers are upfront costs, opportunities, donor-funded projects. So using this as a donor-funded project and where somebody else is in the long run, it's not sustainable, because what happens when the donors stop donating, right? Um, it's going to be a real challenge. I've seen this all over the place. If you're in cocoa and you're in chocolate, NGOs who donate on the farm, they've got a 2-year cycle. If they've got to be successful, and they got to go back to their donors to get money for the next 2 years. And if they don't get enough money, they're going to drop the project after 2 years. So relying on donor-funded projects makes no sense. Local production of starter cultures can make the approach more affordable and sustainable. And this could be interesting, because what it does is we've just created an entire new a category of jobs. So people involved in the cocoa sector can be people with chemistry degrees. We can get investment in terms of laboratories and things like that. That could be interesting for international development, right? But again, is the government going to invest in that? Is the local cocoa federation going to invest in that? Who is going to pay for those things? Not only the setup, but is it going to be a profitable business selling these cultures to farmers? These are economics. And number one, when we talk about sustainability, we need to remember that we need sustainable markets. We need people who are willing to pay money to buy the product. So they have to buy the chocolate made for these things. They need to be willing to pay more for these things. And so in this era where big chocolate companies are looking to reduce the cost of inputs, right, any way they can, right, is anybody looking for ways to make cocoa more expensive? And I think the answer to that question is no. So I'm really interested in the technology. I'm really interested in the idea. And I don't know if you're interested in the idea. If you're watching this, let me know. Go into the comments and let me know. But I think that while this research paper is interesting in many, many ways, and I now have lots more research papers that downloaded into my research library. So whenever I run across anything that I can download, I have a local copy of it. So I've got tens of gigabytes of research papers that I can go and search through. And I don't know that, while does this tell us, I don't know that this research actually tells us anything we didn't already know, right? I don't know it's telling us anything new. If it is telling us something new, right, what it's telling us is that we don't need to replicate, right, a complete copy of the local microbiome. What we can do is we have tools and techniques that we can now use to go and say, here are the yeast strains that we need, and here are the bacteria strains that we need, and here are the fungal strains that we need in order to be able to get the flavors that we want. And here is the temperature curve we should be looking at, and here is the changes in pH. So change in the pulp and the pH growing, so going from maybe originally somewhere between 3 and 5 to 6 or 7, but the changes in the pH of the cotyledon will go down, so 3.5 down to 2.5 or something like that, somewhere in that range. We now have these variables, and we now have the techniques that says we can go and choose, the microbes that'll give us the flavor that we're looking for, because now we have the techniques to do all of the analysis, not just the gathering the data, but also the tools for statistical analysis and correlation between what the data are telling us through machines gathering, right, and sensorial panels. So that's really cool. But again, who's going to pay for this in production? And that's always the case. We're excited about the technology, excited about the approach. What can we learn from them? But what does it mean for the average chocolate lover, right? Are we going to see large companies go and invest hundreds of thousands of dollars to raise the price of cocoa at the farm gate? I don't think that's happening anytime soon. But let me know, what do you think? Do you think this is something that we need to worry about, pay attention to? Are you intrigued with the idea? Do you like the idea of, or did you like going through the paper and trying to understand what the paper is saying? And oh, by the way, the Dia browser is still in beta, But if you are interested in the idea of using DIA, let me know in the comments. What I will do is reach out to you, get an email address privately, and I can send you a link that you can use to go and be a part of the DIA beta. It's a fascinating tool. Again, I find myself going back and forth between a more conventional browser and the DIA browser, depending on what it is that I'm doing, right? And it's been really, really helpful for me. And I hope it's been interesting and helpful for you. But this episode of Pod Save Chocolate, like every episode of Pod Save Chocolate, has been sponsored by members of the Chocolate Life. And so here is the homepage of the Chocolate Life. Let me go there now. It's going to take me a second. This is the homepage of the Chocolate Life right now. I'm running across two browsers. Let me go. Here's the post for this episode. So I've got one or two. I'm going to put one or two references in this, but you can look at the research. Here's a link to the paper that I've talked to, my context, and then this idea about how to go through and read a research paper and some things like that. Talk about what the article doesn't talk about. So all of that is in this post. But if you go and up to the top, there's a little join button, because this episode of Pod Save Chocolate, like every episode, is sponsored by the members of the Chocolate Life. You're not a member, you can go click on the join button. There's always a free membership tier, but if you do have the resources to support the work that I'm doing financially, I don't do advertising. Even if I get to the size of community where YouTube will allow me to monetize this through advertising, I don't plan to do that. I'm not doing Patreon. It's just everything is done through the support of the Chocolate Life on the Chocolate Life. And with that, I want to say thank you much to everybody for joining me today. I know what it is that I'm doing for the next episode, which is there was an interesting article which was published recently in a German magazine about what's going on with Barry Callebaut. And so we're going to go take a look at what's happening with the largest B2B chocolate maker in the world next week, and what that might signal for the future of big chocolate. And with that, I want to say thank you, everybody, for joining me, and to remind everybody, if you're working with chocolate and you're not having fun, you're doing it wrong. Until the next time, everybody, ciao.